there are still people in the waiting room, so let's wait a few seconds to start. So, has the live stream started? Ah, okay, yes. Okay, I see myself. Great. So, this second day of lectures has a program that is very similar to the one of yesterday. So we'll start with Leonardo Pacciani-Mori, who is give the second part of the tutorial on nonlinear dynamics. So without saying anything further, please, Leonardo, you can share the screen and.
Yes. Okay. So you can see my slides, right?
Yes.
Perfect. So hi everyone, welcome back. So before I get started on the second part of this tutorial, are there any questions about what I showed yesterday?
Please, again, if you have any question, please raise the hand with the Zoom feature. You have to go on participants. There are 3 dots, and then you can raise hand. Okay. No one is doing that, so I think you can.
Okay, perfect. So I will start right where we stopped yesterday. So yesterday we were talking about Liapunov functions. So in general, we were talking about how to study the stability of equilibria in nonlinear systems, and I was introducing the use of Liapunov functions as a tool to do that. In particular, let me just repeat Liapunov's second theorem so that we get on the same page we were yesterday. So if you remember, I told you that if we have a generic nonlinear differential equation with x star an equilibrium, and we suppose that in a neighborhood of x star, we can define a function w such that this function here has a minimum on the equilibrium, then the stability of the equilibrium can be studied by looking at how this function w behaves along the trajectories of the system. So for example, if we find out that the time derivative of this function along the trajectory is 0, so if this function is constant along the trajectories of the system, then x star will be an equilibrium stable at all times. On the other hand, if the time derivative is non-positive, so either negative or equal to 0, the equilibrium is simply stable. If the time derivative is strictly negative, so if the function is decreasing along the trajectories, the equilibrium is asymptotically stable. And if the time derivative is strictly positive, so if the function is increasing along the trajectories, the equilibrium is unstable. And then any function w defined with this property here is called Liapunov function for the equilibrium. Is everything clear? Okay, so we talked about this a little bit already yesterday, but I just want to highlight the pros and the cons of using this approach. Now you see that this is actually a very powerful approach because it can basically give us an almost complete information about an equilibrium. And it gives conditions for stability that are sufficient but not necessary, meaning that, of course, we can have that an equilibrium is stable or unstable or anything else without necessarily having a defined Liapunov function. So finding Liapunov function is not necessary for determining the stability of an equilibrium, but is sufficient. The price that we have to pay for having such a powerful tool, unfortunately, is that we can't use it always. So it's not easy to find Liapunov functions in general in nonlinear systems, with, as I told you yesterday, the notable exception of conserved quantities. So if we have a system, if we know that a system has a conserved quantity, generally that quantity is a good first choice for the Liapunov function. Sorry for the ambulance in the background. But if we don't know if the system has a conserved quantity, we can't, we have to use our intuition to find a Liapunov function. So let me show you a concrete example to see how we can use the Liapunov functions. And I want to show that using the lotka-volterra equations. Now, Professor Weitz yesterday in his lecture mentioned that the lotka-volterra equations actually have a conserved quantity. So let me show you how we can find it. So these are the equations of our system. Again, I'm going to do some very simple computations, but they are very non-rigorous. So again, I'm sorry if you're a mathematician. But what can we can do here is basically, let's try dividing the first question, the first equation for the second. So we get dx over dy, and notice that this is not a general method. So what I'm doing here is just trying something and then see if I can find a conserved quantity. This is not like a general technique that can be used always. But in this case, we can rewrite the equations in this way. So here I'm just factorizing x and y. So now that we have written this, we can separate the variables. So I bring on one side everything that depends on x, and on the other everything that depends on y. So you see that we can write dx times delta x minus gamma over x equal to dy alpha minus beta y over y. Okay, so we simply rewrite this as follows. So we divide by x or y on the two sides. So we have delta minus gamma over x equal to dy alpha over y minus beta. But these are very simple functions that we can integrate very easily, because integrating this constant here, we get delta x plus a constant. This here gives minus gamma times the logarithm of x. This here plus a constant, of course. This here gives the alpha logarithm of y plus a constant. And this here gives minus beta y. So in the end, basically we have that delta x minus gamma logarithm of x plus beta y minus alpha logarithm of y, which we call w, is a constant. So indeed, we have found a conserved quantity for the lotka-volterra system. So, sorry, let me just jump here. So again, this is not a general technique. Every time we have to see how our system behaves and do some trial and errors, but in this case, we are lucky. So we have a conserved quantity. Can we use it as a Liapunov function to study the stability of the two equilibria that we know the lotka-volterra system has? So let's see. Let me write this down again, sorry. Okay. So we have the W is delta x minus gamma logarithm of x plus beta y minus alpha logarithm of y. Okay. So in order to see if this is a we can use this function as a Liapunov function, we first have to see if the two equilibria that we know the lotka-volterra system has, which are this one that I'm writing here, are actually minima of this function. So let's compute the partial derivatives of this function here. You see that they are very easy to compute. And so, for example, the partial derivative with respect to x will be delta minus gamma over x, and the partial derivative with respect to y will be beta minus alpha over y. So the first thing that you can see is that these derivatives are not defined in the origin, in this equilibrium here, because of course they diverge for x and y equal to 0. So the first thing that we can see is that we cannot use w as a Liapunov function for the origin in this case. However, you can see very easily that if we substitute the coordinates of this equilibrium here, in both cases we get 0. So this non-trivial equilibrium is indeed an extremum, let's say, for this function. But then you see that if we compute the second derivatives of this function, we get functions that are always positive. So we are sure that this non-trivial equilibrium is not only a minimum of the function, but a global minimum, because it's the only one. So we can use this function, we can try to use this function as a Liapunov function for the non-trivial equilibrium. So what we have to do now is compute the time derivative of the function along the trajectories. So simply, if we compute the time derivative of this function, again, I can just write it down here. You see it's very easy that in this case we have delta x dot over x gamma, y dot, and alpha y dot over y. So in order to see how this behaves on along the trajectories of the lotka-volterra system, we just have to substitute the expression of y dot and x dot from the lotka-volterra system. Now, this is a very simple computation. So you see that if we substitute here, we have delta x alpha minus delta beta xy, minus, here we have gamma alpha plus gamma beta y, plus beta delta xy minus beta gamma, and then we have minus alpha delta x, yes, sorry, I'm for plus alpha gamma. I'm probably forgetting something, of course. No, okay, that's everything. So you see that, for example, this term here and this term here cancel out, then this term here and this term here cancel out, this term and this term cancel out, and then this term and this term, sorry, here I forgot to remove a y, this term and this term cancel out. So in the end, this quantity is equal to 0. Now, we could have guessed that from the fact that we know that W is a conserved quantity of the system, but still, we have computed this explicitly. So if we look at the definitions that I've given you before in the Liapunov theorem, what we have now is that the non-trivial equilibrium of the lotka-volterra system is actually stable at all times. While again, remember that we can't say anything in this case on the origin because this function here is not defined for x and y equal to 0. Is everything clear for now? Are there any questions?
I think we can take the silence as...
Okay. Okay, so let's go on. Now, Professor Weitz yesterday showed us how the trajectories of the lotka-volterra look like, but I just want you to show that again. So for this particular choice of the parameters, you see that indeed we have these solutions oscillating anti-clockwise around this stable equilibrium here. And this is just an example of the oscillations that we've seen also yesterday, where you see that the prey population peaks before the predator population. Okay. So this is more or less how we can use the Liapunov functions to study, the stability of equilibria in nonlinear system. The other tool that I've told you we can use in this sense is spectral analysis. Now, spectral analysis is actually a simpler tool and basically consists in linearizing a nonlinear system around an equilibrium. The basic idea that is behind spectral analysis is the fact that we can approximate a nonlinear system with a linear one if we restrict to, let's say, a neighborhood of an equilibrium. So what we would like to do in general is, given our nonlinear system, we Taylor expand this function f around an equilibrium. So we will have this term here, which is of course equal to 0 because x star is an equilibrium. Then we will have a linear term, a quadratic term, and then all other sorts of terms. And what we would like to do is basically approximate our nonlinear system with the linearized, the linear one, where this J here is called the Jacobian matrix, and is basically the matrix of the partial derivatives of this function computed in the equilibrium. Of course, this is a matrix if we are considering a system in more than one dimension, but if we have that x is just a simple one-dimensional variable, this is just the partial derivative of the function. Basically how this approach works is basically by studying the eigenvalues of this matrix, because if we know this matrix, then its eigenvalues can give us some information about the stability of the equilibrium. In particular, what we can do in this case is given by the so-called Liapunov's first theorem, which basically states the following. If, again, we have a generic nonlinear system and X star is an equilibrium, then if all the eigenvalues of this Jacobian matrix have negative real part, the equilibrium is asymptotically stable. On the other hand, if at least one of them has a positive real part, then the equilibrium is unstable. Is that clear? I mean, I'm going to explain.
There is a question
Okay.
By Mohammad, who asks if this concept is similar to PCA, principal component.
Yeah, I mean, yes, PCA in general is something that you do with rectangular matrices, so matrices which don't have the same number of rows and columns. In this case, however, you always have square matrices because, I mean, if X is a vector of dimension n, of course, also F will have to be a dimension in, a function in n dimensions, and so this will be an n times n matrix. And so in this case, we use directly eigenvalues because we know that eigenvalues and eigenvectors are defined for rectangular matrices, for square matrices. PCA is something that we do in with rectangular matrices, but this is just not this case. Is that clear? Okay. So you see that compared to using Liapunov functions, this approach is actually much simpler because it can be applied to any nonlinear system. So as long as we know explicitly how this function is defined, we can always apply this approach. And this theorem actually is quite powerful in the sense that it tells us that the stability properties of the equilibrium of a nonlinear system, are exactly the same of the linearized system. So we can use this approach to study equilibria in a nonlinear system. The price that we have to pay for having an approach that is simpler and easier to apply is that it doesn't necessarily always give us all the information on an equilibrium. Now, you see already that it can only tell us if an equilibrium is either asymptotically stable or unstable, but there are also cases where this approach can tell us anything about an equilibrium, because you see that if we have at least one eigenvalue with no real part and all other eigenvalues with strictly negative real part, we are neither in these two cases, and so we cannot say anything in this case. What we can do in this situation is just, for example, using other tools, drawing stream plots, or seeing how the equation behaves at higher orders and try to guess something about this equilibrium. Now, I want to introduce another type of stability that we can find in dynamical systems, and that is also relevant sometimes in ecology, which is marginal stability. Now, in general, an equilibrium is defined as marginally stable if it is neither asymptotically stable, neither unstable. But in particular, it is marginally stable when all the eigenvalues of this Jacobian matrix here are purely imaginary. So it is a particular case of an equilibrium being neither asymptotically stable nor unstable, because you see that the case in this third point here is one of these cases, but in this case, we can't say anything on the equilibrium. If, on the other hand, all the eigenvalues are purely imaginary, the equilibrium is marginally stable. Okay, if there are no questions, okay, so let's move on. And let's see a couple of...
there is actually one.
Oh, okay.
Tambin, please mute and ask. Tambin.
Can you give an example of marginally stable?
Yes, I will give you an example in a few slides.
Okay, thank you.
So let's see first a couple of concrete examples.
Sorry, Leonardo, to interrupt. There is another question from the chat.
Okay.
Actually, two. So one is asked by Tuan. So he's saying whether this approach cannot say whether an equilibrium is stable at all times.
Okay. Actually, no. The only thing that this approach can say is an equilibrium is asymptotically stable. This is the only thing that it can say.
And Miguel is asking, what's the advantage of spectral analysis over stream plots?
The advantage of spectral analysis is just that it seems, I mean, you just have to compute the derivatives and evaluate them. So it's something that you can always do very easily, even in very complicated system, in a very high number of dimensions with very complicated functions. Stream plots, I mean, are not always easy to draw, because if you go above 3 dimensions, I mean, I don't know how to draw in 4 dimensions, so it's not, it wouldn't be easy to draw the stream plot on a system in more than 3 dimensions. So this is the advantage of spectral analysis.
Yes. Great.
Okay.
Oh, there is another question by Rasmita.
Okay.
Yeah, hello. So my question is, from the third point, if we get the case that when one eigenvalue is 0 and all others are negative, so can you suggest any method that which we can consider?
Sorry, I didn't hear correctly. I have some problem with audio. Can you repeat the question, please?
Yeah, so question is regarding to the third point, pros and cons.
Okay.
That when one eigenvalue has non-real part and all others are negative real part. So if we arise to a system which shows this kind of nature, then do you know, can you suggest that what will be the next step? How to deal with this kind of system?
Well, it actually depends on the system. I'm going to give you an example again in a few slides where we are actually in this case here. Okay.
Okay.
Okay. So
thank you.
No problem. So if there are no other questions, okay. Okay. So let's see some.
There is another one in the chat.
Yeah.
Does a linear stability implies stability of non-linear?
Yeah, I mean, the result, I mean, the main message of this theorem is exactly this. I mean, that if you have that the linearized version of a system close to an equilibrium is stable, then also the real system, let's say, the equilibrium in the nonlinear system is stable. So what this theorem is saying is that as long as we are close to an equilibrium, we can use we can use the linear stability to induce stability in nonlinear systems. And the same is true also for instability. Okay. Okay, so let's see a couple of practical examples. So I first want to show you again the very first example I've given you yesterday. So this nonlinear system with this cubic function. And just to remind ourselves, this is what we did when we drew the stream plot of this system. So now I want to show you what spectral analysis would say in this case. So now we are in a one-dimensional system. So the Jacobian in this case is simply the derivative. So we simply have to derive this cubic function. And so we get a parabola in general. And then we have to evaluate this parabola in these 3 points here. So you see that if we evaluate this derivative in -1 and in 2, we get positive values. While if we evaluate it in 0, we get a negative value. So in this case, we can conclude by using spectral analysis that 0 is an asymptotically stable equilibrium, while -1 and 2 are unstable. Again, because this is what the theorem says, and in this case, since we are in one dimension, the Jacobian is simply the derivative, and the value of the derivative is the only eigenvalue, basically, of the system in this case. Okay, if there are no other questions, I go on with other examples. Okay. So this is another interesting example. So we have a system in 2 dimensions, x dot equal -x and y dot equal ky to the third, with k a positive parameter. So this is the function that we have to study. Now you see that the only equilibrium of the system is the origin, because this is the only point where both the first and the second components here are equal to 0. Now, let's use spectral analysis. Now, let me write this down. X minus x and ky to the third. Now, spectral analysis tells us, let's compute the Jacobian matrix. So the Jacobian matrix, in this case, if we call these components F1 and F2, the Jacobian matrix looks like this, dyf1, sorry, dxf2, and dyf2. So this is the matrix of all the partial derivatives of the components of this function. So in this case, this matrix looks like -1, 0, 0, and 3ky squared. Okay, so you see that this component here, I mean, doesn't depend on x, while this component does depend on y. If we compute this matrix in the only equilibrium of the system, so the origin, you see that we get this matrix here, which is basically a diagonal matrix with -1 and 0 on the diagonal. So in this case, the two eigenvalues of the system are -1 and 0. So you see that we are exactly in the third case that I've shown you before, in which this system cannot tell us anything about this equilibrium, because one eigenvalue has strictly negative real part, and the other one has 0 real part. So what can we do in this case? Let's try using some tools that we have already seen. So let's try drawing the stream plot of the system. So in this case, the state space of the system is the whole bidimensional plane. And here we have our equilibrium, the origin. So let's try, for example, to see what happens to the trajectories on the axis here. So let me write this down. So you see that, let me rewrite the equations, -x and ky to the third. Okay, so you see, for example, that if we start with x0 equal to 0, so if we start on the y-axis, let me write this here so that it is a little bit more clear. So if we start from the y-axis, basically our system becomes x dot y dot equal to 0 ky to the third. So basically, if we start on the y-axis, the system will always move on the y-axis. And you see that y dot will be positive when y is positive and negative when y is negative, because k, we are assuming that k is positive. So the solutions on the y-axis will move in this direction. Okay. Similarly, if we start on the x-axis, so if y0 in the initial condition is 0, our system will become x dot y dot equal -x0. So if we start on the x-axis, the solutions will move on the x-axis, and in particular, x dot will be negative when x is positive, so the solutions will move in this way, and it will be positive when x is negative. So in this case, the solutions will move like this. So you see that on the axis, the stream plot looks like this. So from this, we can see that actually the origin is an unstable equilibrium, because you see that here the solutions are moving away from this point. Now, if we want to do something a little bit more, we can do exactly the same thing that I've shown you yesterday when we drew the stream plot of the lotka-volterra system. So we can look at when this function here and this function here is positive, so that we know basically the general direction of the solutions of the system. So we don't know exactly with this approach how these curves here move, but we know that they point in this direction. To show you that what I am doing here actually makes sense, this is basically the real aspect of the stream plot of the system computed numerically. So this is a numerical computation of the stream plot. So you see that indeed, in this direction, we have that the solutions are going toward the origin. In this direction here, they are moving away, and so the origin is unstable. And in the four quadrants, you see that the solutions are moving along these curves, but they are pointing in the right directions. Okay. Now, finally, in this case, the origin is also called a saddle point, because when it happens that an equilibrium is, let's say, asymptotically stable along a direction, but unstable in the other, often it is called a saddle point. So is everything clear here? Okay, so let's go on with other examples. Okay, let's see now, for example, okay, sorry, an interesting exercise that I invite you to do, or maybe if we have enough time at the end of this lecture, I can show you, is see what happens to this equilibrium where k is negative or equal to 0. Equal to 0 is a little bit less interesting case, but at least what happens to this system with k is becomes negative. Okay. So let's see now what can spectral analysis tell us when we study the logistic equation and the lotka-volterra system. So in particular, if we take the logistic equation, we have a very simple unidimensional system. So if we want to use spectral analysis, we first have to compute the derivative of the function. Then, for example, if we compute this derivative in the non-trivial equilibrium, so k, you see that we get minus r, which is negative because remember that these two parameters r and k are always positive. And so looking at the theorem that I've shown you before, this means that the non-trivial equilibrium k is asymptotically stable. On the other hand, if we compute this derivative in 0, which is the other equilibrium of the logistic equation, we get r, which is positive. And so we can again conclude that 0 in this case is an unstable equilibrium. On the other hand, let's see what we can say for the lotka-volterra equation. So this again is our system. We do exactly what we did before. So we compute the matrix of the partial derivatives of these functions with respect to the two variables. So this is the partial derivative of the first component with respect to x. This is the partial derivative of the first component with respect to y, etc. Now, if we compute this matrix in the trivial, let's say, equilibrium of the lotka-volterra system, so no prey and no predators, you see that we get this matrix here. So again, a diagonal matrix with alpha and minus gamma on the diagonal. So the eigenvalues in this case are alpha and minus gamma. And since alpha is a positive parameter, we have that one of the eigenvalues is positive. So spectral analysis in this case tells us that the origin is unstable. And if you remember yesterday when we have drawn the stream plot of the lotka-volterra system, we saw that along the x-axis, the solutions were actually moving in this direction. And so we already saw yesterday that this was an unstable equilibrium. Okay, so let's see what happens in the non-trivial equilibrium. So again, we have the same matrix here, but this time we have to compute it in this non-trivial point. And if we substitute this, we get this matrix. Now, this is a very easy matrix. So let's see. Let's compute the eigenvalues. Now let me just write it down: beta gamma over delta and delta alpha over beta. Okay. So just to remind ourselves, the eigenvalues of a square matrix are computed by setting to 0 the determinant of the matrix minus lambda times the identity matrix. So let's do this. We have to compute the determinant of minus lambda minus beta gamma over delta, delta alpha over beta minus lambda, which is lambda squared minus delta alpha over beta times minus beta gamma over delta. So you see that here we have delta and beta that cancel out, and this must be equal to 0. So in the end, we have lambda squared equal minus gamma alpha. So lambda will be plus minus the imaginary unit times gamma alpha. So basically, this means that the Jacobian matrix of the lotka-volterra equations in the non-trivial equilibrium has only purely imaginary eigenvalues. So this means that if you look at what I've told you before, that this equilibrium here is actually marginally stable. Is everything clear here? Okay, perfect. So let's move on. Let's see some other example. In particular, I want to show you this example here. So now we have, again, a cubic function, but a slightly different one. So if we apply also what we've seen yesterday, you see that the equilibria of this function, of this system, are 3, and are 0 and plus minus 1. Now let's use spectral analysis. Well, let's first draw the stream plot of this system. So if we draw the stream plot, you see that we get something. Oh, okay. Yeah.
There is a question by Pablo Lechon. Please ask.
Is a margin stable a stable or an asymptotically stable equilibrium, or is it just a different category?
Sorry, is either a stable or
asymptotically stable?
Okay, yeah, I mean, they are two different kinds of stability. I mean, they are similar but not equal, because asymptotic stability, let me draw this here. So if we are, for example, in a unidimensional system and this is our equilibrium, asymptotic stability means that we know that the solutions are actually moving towards the equilibrium. So if we look, for example, at how the solution behave over time, like this, for example, here we have X star. So if X star is an asymptotically stable equilibrium, we know, it means that we know that the solutions are doing something like this. So they are actually going towards this value. But if we are just saying that X star is a stable equilibrium, this is not necessarily true, and we just need that the solutions remain close to the equilibrium. So for example, if we see that something like this happens, then X is, and we see that the solution never goes away, so it never does something like this, for example, then we know that the equilibrium is stable. So asymptotically stable is just a particular case of stable.
Yeah, and I was asking about marginally.
Oh, sorry. Marginally stable, in this case, basically means that there are, in the definitions that I've given you before, means that there are oscillations. Because from linear theory, you may know that if the Jacobian matrix of a linear system has all purely imaginary eigenvalue, this means that the solution is doing something like this. Without actually tending towards the value. So, for example, if we had like damped oscillations, so something like this would be an asymptotically stable equilibrium. Well, if we had these sustained oscillations that never get damped, then the equilibrium is marginally stable.
Thank you.
No problem. So, okay. So in this case, if we draw the stream plot, you see, again, just to remind ourselves, for example, in this interval, the function f is positive, so the solutions will be increasing. X will be increasing in the solutions. On the other hand, in this interval, the function is negative, the function f here is negative, and so the X in the solutions will be decreasing. So you see that the stream plot suggests that this equilibrium here is unstable, where this and this here are actually stable. Now let's see what we can say with spectral analysis. So here we have a cubic function, so we just have to derive this function and compute it in these 3 points. So you see that in 0, the derivative is positive, and in plus minus 1, the function is negative. So in this case, we have 1 unstable equilibrium and 2 asymptotically stable equilibria. This is the simplest case in which a system can exhibit bistability. Now, we have seen an example of bistability yesterday with the lecture of Professor Staver, but in generally, we say that a system is bistable or even multistable if it has 2 or more alternative stable states. Let me show you how we can reinterpret this problem. And this will probably make things a little bit more clear. So let's rewrite the same system as a particle in a potential. So in this case, we will have an equation of motion like this, so that the acceleration of x is minus the derivative of a given potential. And we choose this potential simply because it's the one that once derived and the sign is changed, gives us the function that we were using before. So in this case, it means that our system is a particle moving in this energy potential. So you see that the equilibria are the minima of the potential, and there are 2 alternative stable states. Now, the interesting thing about bistable or multi-stable systems, as we saw also yesterday, is that external perturbations can move the system from one equilibrium to the other. So, for example, if we again are considering the particle in the potential, suppose, for example, that the particle is in -1. If we give it enough kinetic energy, and by enough, I mean at least the difference between this value and this value here, what happens is that our particle will go in this direction, will override this, will go over this potential wall, and then it will fall back in this other stable equilibrium. And if we do that in the other direction, of course, the same happens. So you see that external perturbations can switch the stable state in which the system lies. And this, for example, we have seen a concrete example yesterday with the savanna and forest switch in a patch of lands. And so this is something that can happen and anytime we have more than one alternative stable equilibrium. Is this clear? Are there questions? Okay. So the last. Okay. Yeah, tell me.
Is this potential the Liapunov function for this system?
Sorry, is the potential?
The Liapunov function.
Yes, in this case, we can use the potential as a Liapunov function, but only for these two equilibria, because you see that in a neighborhood of these 2 points, indeed, we have that this function has a minima in this point, but this is not true. So we cannot use this potential as a Liapunov function for this equilibrium here, because the function has a maximum. Or again, we could do that if we do the trick that we talked about yesterday. So if you remember, there was a question yesterday that asked, what if we define a Liapunov function that instead of having a minimum in an equilibrium has a maximum? Then in this case, we have to be careful that the definition of stability and instability are switched. So in this case, for example, if we find out that the function is decreasing along the trajectories, then the equilibrium will be unstable. But in general, yes, since this is an energy potential, if, for example, we assume that we are in a frictionless system with no dissipation, and so this quantity is conserved, we can use it as a Liapunov function.
So there is a follow-up question. So if this is not a strict Liapunov function, what is the Liapunov function for this system?
It depends. I mean, it depends. In this case, I mean, for these two equilibria, we can use the function as a Liapunov function. In this case, we cannot, and so we have to use other tools. So we can use spectral analysis. We can try to draw the stream plot. So whenever we...
I think that perhaps it's not fully clear that one cannot always construct a Liapunov function.
Yeah, exactly. This is not true in general. I mean, this is a particular case because we know that there is a conserved quantity. If I write any random, let's say, nonlinear function with strange function signs, cosines, powers, exponential, in general, it is not always possible to find a conserved quantity and not even a Liapunov function.
Yeah.
So that's the, I mean, that's the big downside of using the Liapunov function. They can tell us a lot about an equilibrium, but they are not always easy to find.
It's useful if you can find it.
Yeah, exactly. If you can't, you shouldn't lose your sleep on finding a Liapunov function. Okay? Okay. So the last topic I would like to talk about goes a little bit beyond studying the stability of equilibria, and is called timescale separation. Now, this is basically a tool that can be used often to simplify the study of nonlinear systems. Now, let me show you in abstract, in a general case, what I mean, and then I will show you two concrete examples to show you how we can use this timescale separation. So this approach can be used when a nonlinear system can be described by two different sets of variables, xi and yj, not necessarily of the same number, so they can be of different number, and each one of them has their own nonlinear function, which can be different for these two. Sorry? Sorry?
I think was not a question.
Okay. So we have we can have different nonlinear functions here. Now, what happens often, not always, and this depends on our knowledge on the system, is that we can say, for example, that the time scale of these variables is very different from the time scale of this one. For example, if we are describing a large ecosystem where X is the population of elephants and Y is the population of rats, for example, we know, for example, that the reproduction time of rats is much, much faster than the reproduction time of elephants. So we can distinguish basically between slow variables and fast variables in the system. Again, I'm going to give you a couple of concrete examples afterwards. Now, if we can do this, so if we can distinguish between slow and fast variables, for example, let's assume in this general case that xi are the slow variables and yj are the fast ones. So what we can do in this case is, what happens in this case is that the fast variables, so the yj in this case, will reach their stationary values very quickly, since they are the fast variables, of course. So what we can do in this case is assuming that y.j is always equal to 0. Again, let me repeat that in case it is not clear. If we assume that we can distinguish the system between slow variables, the x, and fast variables, the y, this means that as the system changes, the y variables will almost instantly reach their stationary values. So we can assume, we can approximate the behavior of these variables by assuming that y dot is always equal to 0. Is that clear? Okay. So what we can do in this case is basically setting this equation equal to 0. So you see that here we can now invert these functions g here to write the fast variables as a function of the slow variables. If we then plug this expression into the equation for the slow variables, we basically have reduced, let's say, the number of variables so that we can describe our system only through the slow variables. Okay?
There is a question.
Yes.
By Washington Taylor, please.
Yes, hi. Thanks. Quick question. Are you assuming that the YJs have a asymptotic stable equilibrium or something like that? Like if the YJ is oscillating?
I am not assuming anything about the equilibrium right now. I am just assuming something on the speed, let's say, on which these variables change. So it is sufficient that these, for example, set of variables are faster than the other. I'm not assuming anything on the stability or on the existence even of equilibrium.
I see. I guess I'm wondering if the YJs don't have a stationary value. In other words, if they're oscillatory on their own.
Yeah, that is a case in which you probably can do, in which you can't apply this approach, I would say.
Okay, but you might be able to use some mean values or something.
Yeah, you probably can use some mean value, particularly, for example, if these oscillations are not particularly wide, then you can use them.
Thanks very much.
Sorry?
Mas tipo, só você que mexe no negociador, né? Na época era você, Luciano, né?
I don't think I have understood if that was a question or not. Okay.
Yeah.
Okay. So again, let me repeat that this is an approximation that cannot be done always in any case, but it depends on our knowledge of the system. So in the example I've given you before of rats and elephants, I mean, we have to know already that rats reproduce much faster than elephants. In any case, we need some phenomenological knowledge on the system to justify this approximation. So let me show you two examples, practical examples in which this happens. Now, one class of nonlinear system where this approximation is actually done quite often are chemical reactions. For example, let me introduce this nonlinear system. This is called Girer and Meinhardt activator-inhibitor equations. These are nonlinear systems, nonlinear equations, as you can say, and these basically describe the abundance of two chemical substances, X and Y, which are an activator and an inhibitor. For example, to make some practical example, X and Y could be two proteins that either activate or inhibit a particular metabolic pathway or a particular metabolic reaction. So let's assume, for example, that we can tell that the activator is much faster than the inhibitor. For example, let's assume that we know how these molecules are made, and we can tell, we know that, for example, if we raise the temperature of the system, then the activity of this molecule is much larger than the activity of the inhibitor. activator. So we have that X, the activator becomes much faster than the inhibitor. Then what we can do in this case is set X dot equal to 0, so that here we can use this equation to write X squared as a function of Y. Then we plug this into the equation for Y dot, and we obtain this, which basically becomes, let's say, the approximated equation of the system for high temperatures. Because in this case, these variables are the slow one, and so we can describe the system only through this equation, this variable, sorry. On the other hand, let's assume that we know that the inhibitor is much faster than the activator. For example, we know how we know how these molecules are made, and we know that if, for example, we lower the temperature, then the inhibitor, the activity of the inhibitor molecule becomes much larger than that of the activator. Then in this case, we can set Y dot equal to 0 here. So we can write Y as a function of X squared like this. We plug it back into this equation, and we get, in this hypothetical example, the approximated equation of our system for lower temperatures. Is that clear? Okay. So the other example, which is actually more relevant for ecology that I wanted to show you, is the so-called MacArthur consumer resource model. Now, this model basically describes a system of microbial species competing for some common resources. In particular, this here is a schematic representation of the model. We have a set of resources, we have a set of species. We assume for now, I mean, it's not mandatory, but for simplicity now, we assume that the resources are supplied with constant rates. So this SI here are the supply rates of the resources, and that this species here can only uptake these resources. So we are not modeling any other kind of interaction between the species. Now, in microbial communities, there are lots of phenomena that can happen. We can have biological warfare, so species producing antibiotics for other species, or species producing actually resources, so excreting metabolic byproducts that can be used by other species. So we are excluding all these cases, and we are only describing a system where the species can only uptake the resources and grow. Now, if we build the system, it's not necessary for what I want to show you, but if you want, if we have time, I can show you how this model is built. But in the end, the equations of this model look like this, where this here is the set of equations that describes the dynamics of the species population. And this here, on the other hand, is the set of equations that describes the dynamics of the resources concentrations. Here I have just called Ri this nonlinear function. So you see that these are indeed coupled nonlinear differential equations. Now, what people often do, but I mean, this is not something that people always do, is assuming that the dynamics of the resources is actually much faster than the dynamics of the microbial populations. Now, this generally is justified by the fact that, for example, the molecular dynamics that underlies the uptake and the metabolization of resources, which takes place generally in fractions of seconds or few seconds, it's actually much faster than the time scale over which the species grow, because microbial species take at least several minutes, if not hours or days to grow. So in this case, you see that we know from experiments, for example, that we can define this as the fast variables of our system and this as the slow variables. So if we can do that, we can set Ci dot equal to 0 here, and we can write, as you can see, this function Ri as a function of a sum of all the populations. If we plug it back to the first set of coupled differential equations, you see that in the end, we can describe the system by only using the species populations without considering the resources concentrations. Now, an interesting case is basically this model. I mean, there is an interesting case if we consider the same model in a slightly different setting. In particular, these here are the consumer resource equations with two differences with respect to before. Here, I am not writing the nonlinear function Ri that I was using before, but I am just using Ci. But on the other hand, the resources now are not be supplied with constant rate, but I have written, as you can see, a logistic term, a logistic equation for their growth. So if we do this, so for example, I mean, one case in which this could be a good description of the system could be, for example, a system with phytoplankton, where the resources would be phytoplankton, and the populations here would be zooplankton. Of course, we still have to be sure that the other assumptions of the model are true. So the zooplankton are only eating the phytoplankton and not eating other species. But as long as these assumptions are true, we can use this model. If we now assume, for example, that in a particular system, we know that phytoplankton is growing much faster than zooplankton, so we can assume that the CI here, again, are the fast variable. So we can set CI dot equal to 0 and write CI as a function of all the rest. If we now plug this back into this equation and do some rearranging, I mean, I'm not going to show you just because it's just very boring computations and we have only 5 minutes left. But in the end, we get these equations here, where here I'm showing you the definitions of these parameters. This is actually an interesting result because these equations here are called the generalized lotka-volterra equations. These are basically a generalized version of the lotka-volterra equations for systems with more than one species. And so you see that by doing this timescale separation approximation and not considering the equations for the resources, we can actually simplify this model basically to the generalized lotka-volterra equations. Okay, with that, I have finished all the things that I wanted to tell you in this tutorial, so I will be happy to take questions in these last few minutes.
Yes, so we have a few minutes for questions. So I encourage you to ask Leonardo any doubt you might have. Please raise your hand with the Zoom feature if you have any question.
Okay, I hope this means that I was clear.
I will take that as a proof of that, as evidence of that.
Okay.
So, oh, there is a question perhaps.
Okay, great.
Thank you for giving the floor.
Your volume is very low.
Yeah, your volume is very low. I cannot hear you.
Okay, what I'm saying is that if you can come back in the Liapunov theorem, first theorem.
I can maybe if you write in the chat because I really cannot hear what you're saying.
Okay, no problem.
Let me see.
Perhaps while Mohammad is writing the question, there is another question by Pablo. So please ask your question and then we'll go back to Mohammad's question.
Thank you. So I didn't understand very well why. So if the resources dynamics are fast, how can you make the derivative equal to 0? Wouldn't that mean that the resource dynamics are slow compared to the population growth, so that you can consider it constant and therefore the derivative equals 0?
Not necessarily. I mean, what would that mean? Basically, I mean, let me draw this here. Probably this is going to be clearer. So in general, if we didn't make this assumption, let me write, okay, we would have a solution for the populations and a solution for the resources. So in general, we would have that here, I don't know, we would have a general behavior. Let's assume that the populations at a certain point converge to a value. And even here, we would have something like that. So some oscillation, some strange behavior, and then these functions here tend to a stationary value. Setting CI dot equal to 0 in that case means basically assuming that these variables here are so fast that since the beginning they are already equal to their stationary values. So in this sense, they would not change, but they are already in the stationary values. I mean, it can seem like a strange approximation. In most cases, can be a strange approximation, but there can be cases in which this actually is a good approximation. So again, this depends on our knowledge of the system and what we know about it. It's not something that we can do in general.
Okay, thank you. Thank you.
No problem.
So there is a question, Mohammad, wrote the question, and is, when do we have to deal with the Liapunov exponents?
When do we have, sorry?
To deal with the Liapunov exponents.
Again, probably it's not going to be a satisfactory answer, but it depends on the system. I mean, if you see that you have a system where you can easily find a function with all the property that the Liapunov function must have, then that is a case where you can deal with the Liapunov functions. But if you see that you can't and you don't find an easy way to look for the Liapunov functions, and I'm sorry, I mean, it's not my fault, but this is purely driven by intuition, then in that case, it's better to just use other tools. Use linear stability, draw the stream plots, make some numerical simulations. So any kind of other tools that we can use that can help us understand something about the stability of the equilibrium. I hope this was clear.
There is another question by Zenit.
Yes, sir. I just want to clarify my understanding. So basically, we can use time scale separation. Then hopefully we can get to a lotka-volterra equations, and then after which we can just use the second theorem of Liapunov to study the stability.
Yeah. If we are in one of the cases where the timescale separation approximation actually makes sense, yes, we can do that. And I mean, this is something that people sometimes do. They start from a consumer reduced model, then they assume the separation of timescale, and then they work with lotka-volterra equations. I mean, if you want, this can also give you a different interpretation of these generalized lotka-volterra equations, because they basically come from a competitive system. And so, I mean, you can also interpret them in a different light. But yes, I mean, this is something that can be done, of course. And sorry if I continue to repeat myself, if we know that we are in a case where we can do this timescale separation. And again, this depends only on the knowledge that we have of the system. So if we have some empirical, some experimental knowledge that these two sets of variables are actually very fast or very low, or if we have any other kind of intuition that can justify us to do so. Does that answer your question?
Yes, sir. Thank you.
No problem.
There is another question in the chat from Solmas. So she's asking again about the timescale separation. Without the timescale difference, is it impossible to find a single equation for a 2D system?
Yes, if you don't, I mean, for sure there will be particular cases in which you can do that, but in general, if you are in the situation where you can describe the system with two sets of variables and you can't justify the timescale separation, you just have to study the equations with all the variables altogether. Again, there can be particular cases that I am not aware of in which this could happen, but the general rule is this. I hope this answers the question.
Great. There is a question by Dabasmita. Please.
Hello. So I just want to know that what is the broader sense of applying time scale separation. So suppose my system, I know that it can go through it has slow and fast variables, but what is the output I will be expecting after applying this method?
Simply a simplification of the system. So assume, for example, that let's consider this case. So assume that we have a system with 80 species and 50 resources. These are large numbers, but if you compare them with what you can see in natural microbial communities, these are not so large numbers. So if we have 80 equations here and 50 equations here, without timescale separation, you would have to solve 130 different nonlinear differential equations. If you can apply timescale separation, you reduce this number basically to back to 80, because you are eliminating more or less, you are reducing the number of equations from 130 to 80. So the advantage in this sense is simply the fact that you are simplifying a very complicated model, a very complicated system.
Okay, thank you so much.
No problem.
Great. So is there any other question? We have in principle 5 minutes. Yes, there is another one by Sri Rama. Please.
Yeah, thank you for the talk. It's wonderful.
Thank you.
I have one small question in this timescale dynamics. Can we have this, what is that, we can take normal resource consumer system and we assume initially one is faster, another one is slower, and reverse it. Can we get some boundedness on these solutions?
I wouldn't know in general. Probably, again, I'm sorry if, again, this is not a satisfactory answer, but I think that you would have to go in on a case-by-case approach in this case. So looking at actually, for example, how large or how small the parameters are, and see if you can get intuitively any bound in this case.
Because initially, if you instead of solving the original system, can we get the bound for the original system or something like that by using time scale?
I think that would be possible, but again, I wouldn't say always.
Okay.
Again, sorry again if this is not a satisfactory answer.
No, thank you so much.
No problem.
Nonlinear dynamics is full of questions that don't have a satisfactory answer.
Yeah, exactly.
So, is there any other question? Great. So I think we can go in, take a break for 3-4 minutes. We are going to be again randomly assigned to breaking rooms. Feel free to chat with others or to take a break and stretch your legs, get a cup of coffee, some water, etc. So we will be back in 3 minutes. Thank you. Thank you very much, Leonardo.
No problem. Thank you.
Okay, so we are almost ready to start. So we are back on YouTube, and I'm waiting all the participants to get back from the breakout rooms, and then we can start. Okay, great. I think everybody's back. So let me welcome again Joshua Weitz, who is giving the second of 3 lectures today. So please, again, if you have questions, use the raise hand button or ask the question in the chat. And if you're following from YouTube, you can ask a question in the chat of YouTube. So with that, thanks again, Joshua, for being with us, and you can start.
Okay, thank you, Jacopo. I have a different audio today. I don't know if it's okay. Can you hear me?
Yes, yes, I can hear you well.
Okay, good. And hopefully that also works.
Yes.
See the slide?
Yes, it does.
Great. Okay, well, welcome back, everyone, for the second lecture after also your tutorial this morning, and a second lecture in this three-part series that I will be providing as part of this course on virus microbe dynamics, and focusing on, as I mentioned yesterday, on principles, ecology, and therapeutics. And in order to get some continuity, I will do a much briefer introduction, but each one of these is meant to be self-contained, but obviously abbreviate what I covered yesterday, and then go into new material. So just to give folks, again, a context, what I tried to do yesterday was to explain principles of how to think about virus microbe dynamics through this predator-prey lens, and take that forward, not just at the level of the individual cell, but think about the consequences of how a viral infection of a bacteria, a microbe, might lead to the death of that individual cell, but not necessarily the death of the population, and how that could lead to novel changes in ecoevolutionary dynamics. What I will try to aim at today is to think more broadly about the fates of these cells and how that too can change population evolutionary dynamics and even ecosystem dynamics. So that's still ongoing work. And then on Thursday, I will get to the application side, focusing at least on one application, that of bacteriophage therapy. And again, I'm going to try to mix theory and modeling throughout and allude to certain pieces of theory. If some of this is covered in my book, some of it goes well beyond the book, and you can go to the primary source material for reference. Okay, so just to make sure folks have the sort of arc in mind, you have a sense already that virus-host interactions modify the fate of cells on the timescales similar to division times, but that doesn't necessarily mean the death of the population. I've shown, and I'll review just very briefly today, how infection and lysis can lead to lotka-volterra-like dynamics at the population scale, and obviously new dynamics as well. And then these sorts of processes iterated again and again, whether it's in a microbiome or the global oceans, can have potentially large-scale ecosystem effects. And just to remind folks that viruses are embedded, and virus microbe dynamics are embedded as part of complex ecosystems. So the death of, for example, an autotroph or a heterotroph means that those microbes aren't necessarily consumed by grazers, which would allow that organic material to go up to higher trophic levels, but instead are redirected in what is called a viral shunt back into the dissolved organic and particular organic pools, where they can then be retaken up. In other words, shunting organic material back into the microbial loop and keeping organic matter small. So obviously when this starts to happen, as I already described, with hundreds of millions potentially of bacteria per mill, often tens of millions per mill is a typical level, then that obviously is going to compound over time. But do viruses and microbes, I've been focusing a lot on this lytic route in the oceans and elsewhere, do more than just kill or prepare to kill? Are they doing other things? Of course, the answer is yes, and I'll really focus today on this alternative pathway. Okay. So this talk's premise and really the lecture here and a large body of work that we're trying to work on starts with what I would say is, it seems to be a simple question: what is a virus? And when you think about a virus, I already gave some examples yesterday, you often think about the charismatic version. Again, if I had said SARS-CoV-2, you would imagine this particular corona-like virus particle. Here's this virus particle. But I've given some other alternative answers here. This is what is termed a lysogen, in which the viral genetic material is integrated, and here inside the genetic material of that of the microbial host. It could also be extrachromosally and still passed on. And there can be other alternatives, including lytically infected cells, where here we have viruses, but now they're inside cells rather than outside cells. So in some basic sense, although we often, whenever we're ascribing the word virus, we use it really about this virus particle. This particle isn't necessarily the thing under selection. I would argue that this entire set, A, B, C, all of the above are what constitute a virus. And I'll try to get through in today's talk thinking about viral fitness across the entire viral life cycle, whether it's inside or outside hosts. And thinking about viruses in a different way can really lead to new challenges for theory experiments and fieldwork. So I'm going to try to get towards that vista, offering a different perspective, which is much more the parasite host perspective, in 3 parts. I'm going to again ground ourselves in these lytic models via a very brief recap of what I did yesterday, and then spend the bulk of time in parts 2 and 3 talking about mechanisms and patterns having to do with the cellular level mechanism of lysogeny and lysis and how that relates to potential patterns, and then try to explain how we might understand the benefits of lysogeny and strategies that include this alternative pathway in which the viral genome is integrated into the host instead of killing the host that it's infected and releasing new virus particles. Okay, so let me just do this brief recap, and hopefully this will reinforce what I described yesterday. Again, as you recall, we have these bacterial viruses that are diffusing randomly, come in contact with cells, injecting their genetic material into the cell, taking over the cellular machinery, having a self-assembly process, and eventually these new virus particles are released. And as I described already, this is the basis for these essentially lotka-volterra-like models of virus microbe dynamics, in which the death of the host cell does not necessarily mean the death of the population. And just to remind folks, I will be using similar models today. Here I have a simplified system of resources, cells, and viruses, where media is coming in, resources, cells, and viruses are leaving. The cells take up nutrients and then lead to new cellular biomass and new cells. These are then infected, and then with a burst size beta, new virus particles are released. And I've already explained how that can lead to these counterclockwise cycles where we have a large number of available hosts, many viruses which drive down cell densities, leading to decay in viral densities, leading to recovery of cell densities, and so on. And we get these lotka-volterra-like cycles, which we can then see in experimental work. And in this experiment, as I already explained, this is about 10 days between phage T4 and E. coli B. We get these large-scale endogenous oscillations, despite the fact that this is a chemostatic experiment without any variability in the resource supply. So again, the viral lysis isn't leading to the death of the population, but rather oscillations. And over longer time scales, there can be evolution. And you can see here, as I explained the other day, there can be things like cryptic dynamics. And the cryptic dynamics are a result of the fact that hosts are evolving. Okay? So I went through this yesterday and just trying to refresh everyone with the basis for what is often the canonical predator-prey view of virus microbe dynamics. Okay? And so this means in general, as I described yesterday, and I won't go through all the details, so this is the brief recap, which is that in the absence of evolution, we have a notion that virus microbe dynamics should expect to lead to this pattern at the population scale of counterclockwise cycle, which we've seen. But when there's evolution in place, we can get antiphase or cryptic cycles, where you can see the system seems to double back on itself. And I'll just give a note for those who are somewhat new to nonlinear dynamics. This implies that this system must not be two-dimensional, because we can't have it go in 2 directions via the same place in phase space. So we must be missing some feature of the system. And here, of course, that's evolution. We're taking this 3-dimensional system and projecting it down into 2D. And in a co-evolutionary case, there actually can be clockwise cycle, as I went over on Monday. So the takeaway here for the purposes of today's lecture is that population dynamic patterns have been used to infer putative mechanisms, meaning seeing these cryptic cycles, whether it's in predator-prey systems or virus microbe systems, usually imputes the fact that evolution is going on at the same time scale as these population dynamics. but can we take this same approach more generally to non-lytic interactions, right? So I spent a lot of time on Monday on these lytic interactions. Let's change our perspective and think about non-lytic interactions, okay? So now that I just did that brief recap, and I don't know if I should pause, Jacobo, that was really meant as kind of an introduction and summary of what I did on Monday. I don't know if there are any questions that have already popped up, or I can go ahead.
Not in the chat, so if anyone wants to ask a question, please raise your hand.
That's fine. I can keep going if there are more.
There is one by Pablo.
Yep.
Yeah, super quickly. You've said sometime endogenous oscillations. Could you clarify what that is? What does endogenous mean?
Sure. I'm trying to use the word endogenous, E-N-D-O, versus exogenous, E-X-O, to imply that the oscillations are arising because of feedback in the system, not because I'm driving the system with some external oscillator, right? So that's what I mean, that we're driving with a constant input of resources, yet the response is oscillatory.
Thank you.
Good. Okay, so let me now talk and expand our view of these virus microbe systems by looking at alternative outcomes of cellular level infection. To do that, I have to expand our view of the fate of this infected cell, which I've said a few times. We have this infection, and then soon after the injection of genetic material, this can lead to the death of this cell and the release of new virus particles. That's called the lytic pathway. But for certain phage, specifically that are called temperate phage, there's an alternative pathway in which the genetic material is integrated into the chromosome of the bacteria. As the bacteria divide, then the daughter cells also have the viral genetic material, and this integrated form is called a prophage. And later, either by chance or through sometimes a sensing process related to stress or other features, this prophage can be induced, reinitiating the lytic pathway. The key point here is that in this pathway, the lytic pathway, the virus leads to the death of the cell and the release of new virus particles. And the other, there's not a death of the cell. And in fact, even though the virus has, say, gotten or obtained or found this bacterial cell, it is not actively killing it, at least not yet. And this study of lysogeny is also quite old and dates back to really the origins of thinking about both molecular biology and gene regulation, but also in terms of basic virus microbe dynamics. This study has been revitalized in many ways. There's a beautiful book by Potashin, A Genetic Switch, focusing on phage lambda in particular. And a number of studies in the past, I'd say 15 years or so, really have leveraged new techniques to label and look at these same problems, not at the population scale, but at these individual cell or phage levels. And here's an example from the work of Vito Golding, in which they have these labeled phage, so the capsids are labeled, and you can actually see singly infected or absorbing cells, or those in which there are more than one phage absorbing and infecting an individual cell. But they've labeled the system as this capsid with green, but also included a promoter that will express this red fluorescent reporter insofar as the lysogeny pathway is initiated. So what you can see is that some of the cells, and it's not actually a coincidence that this multiply infected cell ends up expressing red, because that tends to be the case, but you can see that some of the cells express this red, and some, as you can see here, are producing new phage, and then they've burst and released new progeny phage, whereas others, as you can see, divide, inherit the property of the mother cell. Okay. What's interesting is that these are not just stochastic outcomes. There is elements of stochasticity, but they're also driven in part by the state of the system. So for example, the viral concentration, or thinking about it in the single cell level as the multiplicity of infection, the probability of lysogeny actually goes up. So counterintuitively, maybe, if a cell is infected by more viruses, this would seem to lead to a more virulent outcome. Would you rather have a large or small dose of a particular infection, you probably prefer a small dose and imagine that the outcome will be less virulent. For temperate phage lambda, and this happens in a number of circumstances, the outcomes are less virulent the more multiple infection, the more phage are infecting the same cell. So here you can see the probability of lysogeny, this option here in which the cell is not lysed, goes up as a function of the multiplicity infection. From here you have 1 to about 20% to something like 80%. So it's not deterministic, it's not 0 or 1, but it actually changes as a function of this multiplicity of infection. And this actually goes back to wonderful work by Philippe Kurilsky in the early 1970s. And just as there is this probabilistic decision, which is driven by some ecological dynamics, so too the induction probability also depends on stress in this nice way. Depending on the UV dose, there's a much higher chance of being induced, whereas depending on the other dose, only a smaller fraction will be induced. Okay, so you can see here that we have these life history traits associated with the fate of individual cells that can vary, and that also can lead to certain feedbacks in the system. Okay, that is the how question. In other words, there's a whole field built around how this works. How does a phage have these alternative outcomes? And today's lecture is not focused on this how question, and you can read more in Patashna for it and recent work by the Golden Group, our group, many other groups have been trying to address this how question, which has to do with gene regulation. But today I want to focus on the why question. Why be temperate? Why not? If this is a virus that's acting like a predator, the simple predator, there's a cell, why not take advantage of this opportunity, kill it, and release many new virus particles? So Frank Stewart and Bruce Levin in a wonderful paper back in the mid-'80s asked this question of why be temperate? And to do so, they also use simple coupled nonlinear dynamic models of the kind that you're hearing about in your tutorials, also I've introduced here, which describe in some sense what happens to cells and to viruses with time. And I'll elaborate on a sort of modern version of these same models, but their premise was one that they call this feast or famine hypothesis, that temperate phage would presumably do better when few hosts are available and extracellular mortality is high. In other words, it would seem, in order to explain this phenomenon that phage tend not to kill, is that if there's very few hosts left, if a phage got in and killed that host, then there would be no host left for its progeny to infect. That is a bit of group or even multi-level selection, right? If it's in the advantage of the virus to do it, why doesn't it just do it? And also they were never able to obtain solutions consistent with it. It was a nice hypothesis, but it was hard to actually figure out a solution for it. So they would introduce these exogenous oscillations, but there didn't seem to a way to actually make a kind of proof or demonstration that this was in fact the evolution rationale for why to be temperate. This is not just a question, and let me just hold here for a moment. This is also consistent with a lot of the phage lambda work in the sense that if multiple phage are infecting the same host, that also indicates there are probably not many uninfected hosts left. And so again, it seems with this feast or famine hypothesis that at that point, hosts may be relatively non-abundant, and if they were to be killed, because that was where the temperate strategy seemed to go, that would imply that maybe the host would go away. But that's not what we find. We find the opposite. This is not just a question of phage lambda, but it also became the paradigm in marine systems, which is known through this moniker of the seasonal time bombs. In other words, lysogeny is prevalent given low productivity and lysis is elevated high productivity. You can see this here in a study looking at the fraction of infected cells during essentially low productive periods and high productive periods. And also shown here is the fraction of bacteria that are lysogens, and they can figure this out by trying to induce them through an inducing agent, for example, mitomycin C. And what you can see is that in low productive periods, there are very few visibly infected cells. Whereas in the high productive, high density environments, and I want to make sure that we separate those two concepts, there seems to be a lot of visibly infected cells. And likewise, there seems to be a switch in which there seem to be many lysogens in other words, phage that are integrated but not actively infecting or producing viruses in the low productive periods, and very few in the high productive periods. Okay? And this became certainly a dogma for many years until a few years ago when an alternative hypothesis was introduced, and that is called piggyback the winner, which is literally the opposite of what I just described to you. And this is that lysogeny should be positively correlated with increases in host density and productivity. In other words, as there are more bacteria and things get more productive, there should be more lysogeny and less lysis, and the opposite, which is, again, the opposite of the phage lambda story and the Stewart and Levin hypothesis. When there are fewer cells and less productive environments, there should be more lysis. And this was meant in part to explain patterns between virus-like particles, viruses, and microbes in ocean systems. This is one particular example where you have viruses on the y-axis, microbes, bacteria, largely on the x-axis, log scale. So we have a straight line here, which denotes a linear relationship. And the data, although scattered, implies a slope that is less than 1. In other words, the ratio of virus to microbes seems to decrease as microbes increase. Okay. So that was a claim, and this was the putative mechanism. So now let me try to disentangle those two claims. First of all, the idea that there is a fixed ratio between virus and microbes, which you'll often see in the literature, and you'll often see it ascribed as 10-to-1, is just not the case. It is a decent median, but we, in a totally independent study, and there's even a third group who did something around the same time, looked at this question and found that here are all these individual relationships by study. And what you can see is that the best fit for the entire data is a power law with an exponent less than 1, implying exactly that the ratio of virus to microbes decreases as microbes increase. But it can be offset. You can have higher numbers, you can have lower numbers, but certainly it seems as if this pattern holds, right, on the abundance side. But the challenge is whether or not this idea is enough to explain that pattern, meaning it's one thing to have the model explain the pattern, but the question, is it unique? Can many models explain that same pattern? In the piggyback the winner model, they used, again, these system of nonlinear differential equations of microbes and viruses, but notably no lysogens. Instead, what they had is that the viral efficiency, so here you have lysis, and the viral efficiency scaled with n over k. So even though the idea is that activity of lysis activity goes down, in fact, their model has it going up. So here you can see this is a standard model, but it goes the other way. It's true that if you look at the outcome of the model, it finds this pattern, and this is the result of our reanalysis of this work. But it is also true if you just vary parameters because of parameter entanglement in the steady state outcomes, you can also get this feature that the virus-microbe ratio goes down as microbial density goes up, and you don't need to invoke this mechanism in order to see the same relationship. So that implies that this pattern alone may be insufficient to make the claim that this is a piggyback the winner mechanism. However, they then looked directly at metagenomic evidence and tried to see in the viral fraction, so those are virus particles outside cells, what fraction of them had things indicative of provirus-like reads, in other words, indicating that there may be the possibility of being temperate, and claim that this went up, which is what their hypothesis is. This is our reanalysis of this data, and as you can see, there's not much of a relationship. In the provirus-like reads, in the integrases, and the excisionases, all these are hallmarks of being temperate. And in our view, neither the metagenomics nor the abundance data support directly this evidence of piggyback the winner, but notably, it doesn't go down either, right? So there seems to be an absence of evidence for a particular relationship. Nonetheless, this idea that one can go from a pattern to a mechanism is enticing. And in fact, the following year, a separate study concluded that the findings of a decline of a ratio of viruses to microbes, here I'm showing again, virus to microbe ratio, microbes, you'll notice again and again in this separate metagenomics study of linking viruses to hosts that it seems like this pattern is ubiquitous. And because that was claimed to be a proof of piggyback the winner, they say it corroborates the proposed theory. Having already worked in this space a little bit, I saw this paper and was a bit concerned. And again, maybe if we were together, I would ask to see if you were concerned, but you'll notice that these lines, which denote virus microbe abundances or metagenomic inferred abundance for different virus host pairs, all seem to have parallel slopes. I hope that's clear, right? And if you can look at it, you'll see that if we started about -2 to 2, something about this would be a slope of -1. And I was worried that these parallel slopes all looked a bit too much like -1. Here is our replot of the same data with a -1 slope as a guide to your eye. And you can understand that this y-axis is not something independent, but it's a ratio of virus to microbes plotted against microbes. In other words, this is a plot of y over x versus x. But if y over x goes like x to the -1, this implies that if we just look at the virus abundance to the host abundance, you can see that almost all of these lines are statistically not significantly different than 0. In other words, an absence of a slope, which means that it appears to be unrelated to host abundances, or at least we don't have the evidence say they're related. And frankly, that's a counter indicator for piggyback the winner, which implies viruses are going up in density as hosts go up in density, just not as quickly, which is why the ratio goes down. So again, the takeaway here is that we feel there's an absence of evidence for positive correlation, whether looking at lysogeny proxies or examining ratio of virus to microbes as a proxy, but it does raise some interesting questions because we don't see the opposite relationship. Right? We're not seeing the sort of inverse relationship as one might expect. So I'll try now in this third part to provide some thoughts about how we might think about the benefits of lysogeny and when we might expect to see more or less of that strategy, both in experimental systems and natural systems. And now is probably a good holding point. Let me get one thing here before my clock was on the wrong side of the room.
So yes, so if there is any question, please. Okay, there is a question from Leonardo, please.
Yes, hi, can you hear me?
Yep.
Okay, thank you, Professor. This is a very interesting lecture. I was just curious about what are the mechanisms that trigger lysis from lysogeny? You said that stress and sensing could be factors, but I was just curious if you could give some examples on when do cells with the viral genome included in their own genome start the lytic cycle.
Right, so there is a regulatory mechanism that keeps a prophage stably integrated inside a cell. And it's true that we know about some of these largely from a few model systems, so I'm sure there are many things left to discover. But the idea is that, for example, if there is DNA damage to the cell, then there's a repair mechanism that is going to be activated. It turns out that the repressor system, which keeps the prophage stably integrated, can then, those repressor proteins can then be cleaved by this same DNA repair mechanism, which then leads to an induction event. So a typical way to think about in natural systems is DNA damage, some sort of damage agent or stress, whether it's UV or mitomycin C, are all experimentally standard ways to induce because you're leveraging this sort of evolved system. There may be other mechanisms when we think about the way out. There are really two decision switches there, in and out. And going in, there's all sorts of interesting questions to be raised, which I didn't go into today on the how side, because if we have multiple phages that are affecting the same cell, in some ways we have a gene dosage question that we need to ask, because now you have more copies of this phage genome, and you might think, well, how would that change an outcome? But these are systems with nonlinear feedback. So if we're increasing the rates of everything, but there's a nonlinear feedback, we can actually preferentially end up in one kind of fate rather than the other. But from the induction side, there's also a switch. And again, as I said, that is controlled by a coupling between the regulatory mechanism, for example, the C1 repressor, and the status of the DNA repair enzymes, and those two together can lead to induction. There may also be cued into quorum sensing, and there are other systems too. For example, the arbitrium system, which is quite interesting from the SOREC group, and that has its own mechanisms of integrating in those decision switches. So I think as we begin to see more of these model systems, we'll have more of these examples.
Okay, thank you. Thank you very much.
Great. Any other question? Oh, yes, there is a question by Ravi, please.
Hi. Just a quick question. Are there any fitness costs while the virus is inside, the phage is inside the before lysis? Are there physiological costs to the bacteria or anything like that?
So I will be talking about some of the ways in which there could be fitness benefits and fitness costs, right? Because certainly a fitness benefit, if the virus brings with it a gene that is somehow beneficial, then in fact, there's an entanglement, and the reproduction of the cell is also reproducing the virus, and it's sort of in their mutual interest. And I will talk about that next. There could be costs, though it seems, given the size of bacterial genomes and viral genomes, relatively speaking, that replication cost may be small, but there may be other costs with respect to the expression of repressor proteins. There may be other costs if they're bringing in genes that are not beneficial, right? So there can be all sorts of ways.
Yeah.
I'll talk more about that in the next section.
Yeah, sounds good. Thank you.
Great. There is another question by Miles.
Yes. Hi, Joshua. Just a clarification. So basically that last slide that you showed us, what I understood is that virus abundance is essentially invariant to the abundance of host.
In this particular, and let me clarify, in this particular study, which tried to use a metagenomic-based approach.
Right. Okay.
Whereas if we look at direct particle counts and microbial particle counts at the population level, at the community level, excuse me, then we see a positive relationship.
Yes, but sub one.
Correct.
Okay, thank you.
Yep. Should I continue?
Yes, I think.
Okay, good. And I think I'm still on track, on time. So it's looking good. So let me go back and ask this question again, because despite the disagreement and some of the debate, and I want to point out that debate in science is usually a good thing, right? We're trying to clarify ideas, and we have an intent, a shared intent, to try to increase our own understanding, shedding light on complicated, hard problems. And so I think this question of what environmental conditions should favor lysology rather than lysis is a good one. And I can imagine there are circumstances in which there may not be one monolithic answer to this question. But nonetheless, I'm going to try at least build a conceptual foundation for how we could begin to address it. And I'll try to do this by making an analogy. And I realize this is a very international group. So in English, there's a saying, a bird in the hand is worth two in the bush, which I will try to translate because certain times sayings may not always be immediately understandable. But I hope you get the idea that you have something, it's in your hand, and you might be willing to trade it for something uncertain because it's out there. But if you're going to do that, you better get more because those things are uncertain. I'm not going to trade my bird in the hand for one in the bush. I should get at least two. And certain cultures have similar sayings. I was told in Spain, there's a similar saying, but a bird in the hand is worth 100 in the bush or something similar. So you can tell me about uncertainty in cultures and the number of the trade that you have to make. But I will try to make a new puzzle here that a virus is in the cell and it's worth N in the bloom, meaning the virus is inside the cell and you can put out all these virus particles, but what is N? And also, how many cells is it or need to be out there, and how uncertain does the situation have to be for it to be worth giving up the cell that you found that the virus has found, and then going back out into the environment, this uncertain environment. Okay, so to do this, I'm going to move away for a moment from the population view and take an individual view. And again, I've already described this notion of this encounter, and I'm going to use and anchor our thinking in terms of a lytic cycle in which we start with the virus that injects its genetic material into the host, but I'm going to call this the mother virus. It lyses, leading potentially to hundreds of virus particles, but I will claim that this is not the fitness that we should talk about. Because many of these, if not most of these virus particles, maybe they're defective, they don't ever find a host. Only a very few, in this example, 3 end up infecting a new host, and we'll call these progeny viruses. And I would call the fitness at the individual level here as R, HOR for horizontal, as 3. Okay. And in contrast, if I think about temperate phage, yes, they can do this, but they might instead integrate. You might ask the question, well, why? There's no production of viruses. Viruses are these particles. How can you have fitness? And how do I account for the fact that a strategy is avoiding making itself, right, this virus particle, if that's what we think of viruses. But instead, if I think about the life cycle as being under selection, then here we have a mother virus. It divides 3 times. Each of these have their own fitness. These, we can arbitrarily call these daughter viruses as progeny, and eventually this cell dies. So here, by initiating lysogeny, I would say that the fitness at the individual level of this virus is 3 new progeny viruses, and I've called this VER for vertical, and yet no virus particles were produced. My point here is that it seems that 2 vastly different strategies can have the same fitness at the individual level. I put that in quotes for intentional reasons. And how does this depend on cell densities, on the environment? Because we want to connect this back to the ecological context. Okay. So let's go revisit this population dynamics of lytic viruses. I've given you this individual perspective, but we can think about the population perspective as having a population of susceptibles, infected, and virus particles. Susceptible cells grow logistically, they're infected, lead to new infected cells, which over a time period 1/8 of this latent period lead to a lytic event. All of these things may die or decay, and we could get essentially conditions by which a susceptible population may be infectable by a virus such that the viruses increase in abundance and proliferate. And this is nothing other than looking, again, as I mentioned before, about essentially searching for a positive eigenvalue of this otherwise this stable system in the absence of viruses, but unstable given a small introduction of viruses. And you can see here viruses should increase in population within these cells using exclusively horizontal transmission, only through the lytic pathway, whenever this criteria is met. And again, I don't want to delay my interpretation. I've chosen this calligraphic R because I mean intentionally to evoke this notion of a basic reproduction number. And now, since everyone is an armchair epidemiologist, some of us are actually doing this kind of stuff, but you will obviously recognize what I mean by that and why the condition is greater than 1. And let me unpack this. Let's take the perspective of an infected cell. An infected cell will lyse eta over eta plus d prime of the time because it could either die via some other mechanism or through viral lysis. So a fraction of an infected cell, a fraction of the time, it will lead to a burst event producing beta viruses. Of these, only a fraction will infect cells, which, as you can see, depends on cell densities. Alternatively, I could start with a virus particle, and starting with a virus particle, ask how many virus particles are produced in this life cycle. Well, a fraction of those virus particles, right denoted here, will find cells. Of those, a fraction of those infected cells will burst, times the number of new virus particles produced. And either way, when I think of a life cycle from in a cell sense or in a virus particle sense, I get this same number. And again, I've written it here out in words. When this is greater than 1, we have the increase of this virus population. Notably, though, it is not inevitable. It depends on the susceptible population. Because as you can see here, the chance of actually finding a host is going to saturate as we increase susceptible populations, obviously go down as we have fewer cells. So if we have a fixed burst size, it means that you have to have a sufficient number of susceptible cells for there to be invasion. And here I've written out the R0 criteria. Here's this breakpoint. And below this point, this virus would be unable to invade, even if it has a very high burst size. Right? Again, showing you that it's not just about how well the virus exploits the cell, but the ecological context. And clearly, in all of these cases, as we add more cells, this seems to be enabling a larger state space of potential invasion scenarios for this horizontally transmitted virus. So the takeaway here is that ecological conditions with more susceptible cells and viral traits with more efficient infections favor this lytic antagonistic mode. Let's now do the same thing for latent viruses, including temperate phage, and focusing again only on this exclusive route in terms of our calculation. But here we have susceptible cells, lysogens, which can also divide, and I'll just note that N is S plus L, so it's the total number of cells in the system. And here we have infection, but some of these cells may lyse, and this fraction P, and another fraction Q may be dividing. And when there's a lysis event, we get production of new virus particles. Okay. I'm going to focus on the case where P is 0 and just look at this exclusive growth via lysogeny and ask the question, how can viruses increase and proliferate in this population? They can do so using exclusively vertical transmission when this number is greater than 1. And we can think of this as essentially the average number of new lysogens produced, given one lysogen in the population. And to do that, we see that the lysogens will persist on average 1 over d prime, which is why we have this 1 over d prime. And they will have a division rate, b prime, and it'll modulate it by density. So if we have a rare lysogen and the susceptible cells are at high abundance relative to their carrying capacity, it will be very hard to proliferate. So you can see this is in essence division rate times cell lifespan. What this also implies, and this goes back to a question that was anticipated near the end of the second part, is that the benefits or costs, the direct benefits or costs, are going to modulate the conditions by which this vertical transmission pathways could be favored. When the susceptible population is relatively low compared to this carrying capacity, then you can see, depending on the benefits, this B prime relative to B, if there are high benefits and even low fitness benefits, there's a large regime in which this rare lysogen can proliferate. But when there are fitness costs, well, it doesn't ever seem to be good. It can't ever do it alone. Notably, that irrespective of the fitness benefit or cost, you can see that this R0 level is going down, so there can be a transition depending on the level of fitness benefits, at which point low densities are actually more likely to yield the proliferation of a lysogen rather than high densities. So now we're getting some direct evidence, rather than having to appeal to this idea that phage should not kill their host because that would be bad for them in the future, but rather saying it's good for them now. As you can see here, if these viruses were to try to kill these hosts at low host densities, this is not a viable strategy. Whereas it can be a viable strategy to forego killing a host and proliferate along with it as a prophage. So the takeaway here is that ecological conditions with reduced niche competition, direct cell benefits, or low variance survivorship can favor latent strategies. We can even start to do intermediate cases. There are not just these dichotomous outcomes, but also other forms of infections, including chronic infections. And most notably, this is through filamentous phage, which bud rather than burst, in which case these infected cells are dividing, but you'll notice that there's a production rate of viruses from infected cells, and that doesn't include the death of the cell. And here in my individual example, you'll see that this cell divides once, but buds twice, it has more than 2 budding viruses, but 2 of these budding viruses infect horizontally. So we have a basic reproduction, which is the sum of the horizontal and vertical components. And so when we actually do the analysis in full, we identify precisely the sum for the reasons I've just explained. That we have this vertical average number of new infected cells vertically, and here there's an average duration during which there's alpha new viruses produced, and there's a fraction of them that find new cells. So we have the same rationale. When we put all of this together, you can see that these intermediate cases, although this is a chronic mode, I'll go explain the temperate version in a moment, can end up being invasible across the entire spectrum of densities, where these dichotomous outcomes will eventually fail at these other extremes. In this particular case, we have the highest basic reproduction number in the middle, but I want to point out that that is not Malthusian fitness, so we have to think carefully about feedbacks, which is what I'm going to start talking about in a moment. Okay, so let me just do this takeaway, and then maybe there'll be a few questions before I get to the final part here. Just to say that, again, that these temperate or chronic modes can be favored when susceptible populations are low. So now we have really beginnings of answers to why be temperate. Because on the other hand, this virulent strategy, it's not just that it's bad for the host population, it's bad for the virus. It's not evolutionary beneficial to kill the host because you simply don't propagate. There are not enough susceptible cells in order to produce more than 1 infected cell on average. And this means that this range in which this non-horizontal mode is favored could be much greater range than we have expected, and suggest that we might find this in far more cases than we've thought about before. So to answer this question, I think requires a unified metric, at least to answer the question of invasion. The long-term question remains open and a challenge for the field. Thinking about virus-host dynamics, if we want to understand integration, I think we have to go beyond the predator-prey dynamic, because you can't account for it. There's not an apples-to-apples way to compare. But if you use this epidemiological notion and think about this parasite-host infection as transmitting horizontally or vertically, we do have a unified way that we can compare temperate strategies with these lytic or virulent strategies. Okay, so I should pause there before I do a few more slides and wrap up.
Okay, so there are a few questions from the chat, so perhaps I can start reading them. So the first one is from Egar and is the following. Is there any impact of microbial viral coevolution to the lysogeny level of the phage? And is there any benefit for the microbes to keep the viral gene kept integrated to their genome?
Right. So yes, there is coevolution. There's been very limited work on the evolution in the lab of lysogeny. In the next stage, I'm going to talk about some of this within temperate phage, and also to point out that one of the benefits of having a prophage, not only can there be the shift of genes, though of course the danger for the virus if it doesn't get out is that most of its genome is under neutral selection, which then can drift away into inactivity. But also an active prophage can protect I don't think that's my computer, can protect a host against infection by certain kinds of viruses. So it's not universally protective, but there can be things like superimmunity. And in other words, it helps protect the virus from the bacteria from infection by the virus. That is a benefit.
Great. There is a second question, and is, are the progenies from lytic cycles identical? Because obviously the progenies from lysogeny would undergo several mutations, especially as they cause plasmid degeneration.
Well, people have asked this question, are any 2 snowflakes the same? Right? Each one is a little bit different. I think it's a fair question about viruses, both in terms of their assembly, obviously they're going to be slightly different, but in terms of their genome, you can do the math and you ask the question with something about 50,000 base pairs and an error rate that's typical for double-stranded viruses. I imagine that maybe some of them have precisely the same genome, but many might not. And so the lytic cycle, and I have to think through that a little bit more carefully. I want to be careful about whether or not I really believe that. Let me think more about that, whether I think that there, on average, you expect differences. You probably do, but remember that there is an error correction mechanism. So if it's going through a few generations, we can ask the question, is that going to be any different than maybe a slightly higher rate in the replication cycle during lysis? But yes, you do expect that the progeny are slightly different, and that's the grist for evolution that can happen for the virus, both because of the accumulation during the integrated phase, but also in the event of the induction during the lytic production phase.
Great. There is one last question, which I think is going to be short. So is there any assessment on how many of the phages are temperate?
Are what?
How many are temperate?
How many are temperate? Meaning, how, if we go out to a natural system and we were to ask the question, is that virus that we've identified have the ability to be temperate? Is that the question in some sense? Yeah, I don't think we have a great answer to that. That's in fact the interest of a number of groups. It's hard right now to figure out, in some sense, the potential or conditions where something may be temperate. There are certain hallmark genes. People have been working on this the past few years of looking for omics-based methods to identify hallmark genes associated with lysogeny. It doesn't mean that that's going to be fulfilled in a particular system. There may be other mechanisms. So I think we probably have an undercount, and it's probably more ubiquitous than we expect, but I don't have a fraction that I can say that fraction in natural systems is absolutely temperate and the other fraction is not. I mean, the absence of integration excisionase are probably a good sign, but we're again still looking for the things we know. I don't have a good number yet as a firm estimate.
Great. So there are no other questions.
So let me just wrap up here. I only have a few more slides left. Yeah. Okay. So the problem when I've given this notion of this initial invasion. The problem, though, going back in time is that the models are actually quite different. Frank Stewart and Bruce Levin's model included resources, but didn't include an explicit infected state. There's another model, which I'll talk about, which has lysogens, but again, doesn't include some of the other features like resources. We've built these more complicated models in which there's an active switch between lysogeny in this active state and an active state of induction with or without resources. So it gets to be a little bit confusing with respect to whether or not these findings hold. And this means that you really have very different model structures, or what appear to be different model structures, and how much of what we just found transcends these details. I will just point out that we think that this does transcend details. And in a paper that just came out a few months ago, though who knows how many months ago it was, because this year has been very long, so I've lost all track of time. We have found the same structure for R0 here, for all of these models. And I've shown a diagrammatic view, which I know this looks crazy, but we actually can use this to calculate. And I'll just point out that the basic reproduction in all of these models can be written in this diagrammatic form, where we have essentially loops, which are a lytic loop, a lysogenic loop, and what we call a lysolytic loop. But notably, the form involves things with a square root, which I will also note have been observed before algebraically, because you get these conditions and are noted as complicated. What we've interpreted these as the first generation and second generation loops. And because R0 is the average number of infected cells produced in a generation, here when we take the second generation, we have to discount them by a square root because we're essentially multiplying these together. I'll also note that it gets discounted by impossible second generation loops where we hop from one lysogenic loop to the lytic loop. It turns out that when we actually want to calculate these things, we can use this formula, but of course, the values here are simply the basic reproduction number of these horizontal and vertical loops And those will depend on the model details, but the qualitative form is precisely the same. Okay, so you can write crazy formulas. I won't make you, just to point out that you can actually do this for all these models, but they all reduce, even though the algebra is complicated, to the same fundamental form. Okay? And what this means is that we end up getting the same kind of answers, whether we look at the implicit model of infection or the explicit model. And the takeaway point is that when prophage provide a benefit to cellular growth, what we see is that this vertical strategy is always good. When they impose a cost, it always seems to be bad. The horizontal strategy obviously gets better as there are more hosts available. And there are intermediate strategies, as I mentioned before, that can get the kind of best of both worlds and have a positive invasion fitness irrespective of cell density. But we still raise this question of what happens here at low cell densities, because it seems like then we could have a problem where there are regimes in which none of these strategies seem to work. As you notice here, none of them seem to be invasible. And just to point out, different model structures, some quantitative differences, but qualitatively the same answers. To think about this really does require a long-term framework. And I know I'm at the end of my time, just to point out that Berngruber and Gendon, in a beautiful paper from 2013, analyzed this by looking at the ratio of these virulent to non-virulent types, showing that there was evolution happening in terms of these strategies at the time scale of the epidemic, right? So that you have changes in the ratios, just like I alluded to in my first lecture, at the time scale of invasion. But let me try to unpack it here in a different way. What we've done here in this simulation is ask the question, can temperate viruses that impose a direct fitness cost invade a system in the event that they have an indirect or ecological context benefit, which is they provide immunity to infection by lytic viruses. So they hurt the host in terms of growth, but help in terms of immunity. What we see is that when we start the system, here are cells, here's the resources, we add these temperate viruses, they go away, they can't invade. There's many hosts that are available, but actually this favors the rare type that imposes the cost on growth. But this is an ideal time for lytic viruses to invade. So if we add them at some low densities, they immediately shoot up, reducing cellular densities. And now we re-add the very same temperate virus that we tried to add before that failed, it can invade. It's competing with fewer cells. And also it's protecting those cells that are infected from infection and lysis. And what we find is that depending on the probability of lysogeny, these different colors denote the degree of immunity, no benefits, and complete protection from infection. We see that as long as there's some benefit of infection, we have a temperate phage that can invade when it's invading not a fully naive population, but one in which lytic virus are already there, which implies that in some sense, lytic viruses can help temperate viruses invade, even if they impose a fitness cost in a growth sense, but not insofar as they provide some contextual benefit. What I hope to take away from this is to say that if we want to think about the long term, we're going to have to think about coexistence, not just of viruses and hosts, but of different kinds of viral strategies. And there may be cases where, in fact, these lytic virus strategies make a template and restructure or reshape the environment so that temperate and lytic or virulent strategies can coexist together. So to close, and I think I'm just at time, what is a virus? I would argue it's really all of the above, that we should be thinking about viral fitness across the entire viral life cycle. And in doing so, I hope that we get some new ideas, not just for theory, but also for ways to probe invasion and experiments and interpret, including really at these marine systems, we're finding more evidence of non-lytic infections and trying to understand both when they arise, how they interact with these lytic infections, how ubiquitous they are. These remain truly open questions for the field. And with that, just to point out, a lot of this has come out in the past few years, goes well beyond what is described in this book. And I've given you just a few examples of ongoing work, and happy to take some questions before you go into your break.
Great. So thanks, Joshua, for the fantastic talk. So if there is any question, please write it in the chat or raise your hand. Any question?
We took a number during the talk, so
yeah, I think it's fine that we. If there is no, okay. So I think we can stop here. And Joshua, oh, there is a question. Okay. So please go ahead with the question, Monday. Please, if you want to ask the question, Monday.
I think you are muted, Monday.
Yeah.
Maybe you can type the question into the chat because we can't hear you. Or you can write to me afterwards. I think we're having a technical difficulty, Joshua.
Yes. Okay. So I mean, Joshua will be with us in 2 days from now. So if you have questions, you can also ask the day after tomorrow. So thank you again, everybody. So we'll resume tomorrow at the same time as today. Okay. So keep in mind to always check the program because there are changes. And thanks again for everyone. Bye-bye.
Bye-bye.